Hi. In this mini lecture, we're going to take a look at the last cellular component of the blood, which is our platelets, and the process they participate in, which is the blood clotting process. So first, a look at a platelet. So we've seen them under the microscope. So here's our platelets, these little fragments, and the unique thing about platelets is that they don't actually function until they are dead. So they serve their purpose in our body once they have been deceased. They come from a living cell called a megakaryocyte. So you can see one here that when it dies, it breaks apart and it becomes the fragments that enter our blood and become the platelets. So their function is in the blood clotting process. So if we look at where they come from, so we have our stem cells, right, in the red bone marrow, the hemocytoblasts. They differentiate in two types of cells. So platelets are going to come from the myeloid stem cell line. Myeloid stem cells differentiate into a cell called a megakaryocyte. So this is the living version of the platelet. When this megakaryocyte uh, has fragments that break off, those are what are actually becoming the functional platelets that we find in our blood. So the process of stopping bleeding is called hemostasis. So we're going to take a little more kind of in-depth look at the steps of hemostasis, but this picture kind of summarizes the basic steps. The first step, when we have a break in a blood vessel, the first thing that happens is that smooth muscle, right, that makes up the tunica media of our blood vessels begins to contract, and this causes what are called vascular spasms. And vascular spasms from that constriction of those blood vessels are going to help slow the blood loss. So that's our first step. Our second step, which really kind of is highlighted in both of these pictures, is the formation of what is called a platelet plug. So the platelet plug is going to form from the adhering of platelets to the site of the break. So when we have a break in a blood vessel, you can't see them here very well, but we'll draw them in. We have fibers of collagen that get exposed, and the platelets stick to these. So as the platelets are flowing through in the blood supply, they're going to be sticking to those little collagen fibers. And then when platelets get stuck there, they release chemicals out into the bloodstream that are going to attract more platelets. Okay, so this is an example of a positive feedback mechanism involved with our body. So more chemicals, more platelets, more chemicals. And then the third step of the stopping of bleeding of hemostasis is the actual coagulation. So coagulation involves clotting factors. So within the blood plasma, there are going to be clotting factors, and we'll take a look at them in more depth in a few minutes here. And they interact with calcium ions and form a chemical called thrombin, which then is responsible for taking another chemical called fibrinogen and converting it into a molecule called fibrin, which catches more platelets and forms basically like a mesh. So it's a mesh of proteins and cells that then seals off the break within the blood vessel. So that's kind of the overview of the steps of how blood clots form. So now we're going to take kind of a little more in-depth look at clot formation and at some of the clotting factors and chemicals that are involved in this overall process of stopping blood.
Hemostasis is the normal physiological response to limit blood loss after vascular injury. Blood clots are made up of activated platelets, red blood cells, and fibrin, which hold the clot together. The process is mediated by around 30 proteins, which are known as clotting factors. Platelet activation and aggregation begins when vascular injury exposes collagen and von Willebrand factor from within the vessel wall to the blood. Platelets adhere to these proteins and become partially activated, changing shape and releasing signaling molecules which recruit more platelets to the site. At the same time, a protein called tissue factor triggers initiation of the coagulation cascade, the process that leads to full activation of platelets and the generation of fibrin and clot formation. Vascular injury causes tissue factor on the outside of cells in the vessel wall to come into contact with factor 7A and factor 10A in the blood, leading to the production of a small amount of thrombin. This thrombin fully activates platelets and triggers a cascade of clotting factors that activate each other, culminating in factor 10A-mediated generation of a larger burst of thrombin production on the platelet surface. One molecule of factor 10A leads to the formation of 1,000 molecules of thrombin. Thrombin converts the soluble protein fibrinogen into insoluble fibrin strands at the site of vessel injury, while at the same time activating factor 13 protein to cross-link the fibers. The fibrin mesh stabilizes the clot and traps more platelets and red blood cells, facilitating its growth. In normal physiological conditions, the process of clot breakdown or fibrinolysis begins as the structural integrity of the vessel wall is restored. Endothelial cells begin to secrete a protein called tissue plasminogen activator, which activates the protease plasmin to degrade fibrin, dismantling the structure of the clot and restoring the integrity of the vessel.
Okay, so to kind of recap the steps here, so that first step, which really precedes the formation of the clot, remember, was vascular spasm. So here we can see a blood vessel being cut, and then we can see how the reduction in the size of the lumen, right, from the contraction of those smooth muscles results in a smaller lumen, right, which is going to slow down the blood loss initially until we can get the site repaired. So the formation of the platelet plug itself, here we have our damaged tissue, right? So this is going to be inside the vessel. This is going to be outside the vessel. So we have damage that has traversed the tissue here. We can see the platelets, right, beginning to accumulate at the site of the break, right? They're going to be attracted there by the fibers, those collagen fibers that get exposed when we have a break in our tissue. And remember, they're going to release chemicals, right, that attract even more platelets to this area. And then here we have a little more detail on the steps of coagulation. So we have two pathways. We have the extrinsic pathway, which is going to require factors from outside the blood in order to be able to be triggered, in this case, the damage to the vessel. And then we have what's called the intrinsic pathway, which is going to involve factors that are from within the blood. So the extrinsic pathway is going to, so this is going to be triggered by basically like tissue trauma, the events of this. And this pathway is going to react faster. And then there are chemicals that are released intrinsically from the cells involved here. And this is going to trigger the intrinsic pathway, which is going to respond more slowly. So this is going to take typically somewhere between 1 to 6 minutes for the blood to begin to clot. But these two pathways work together and act on the common pathway. So, for example, if we have damage to tissue, right, we're going to get the platelets starting to aggregate here. We get the release of a clotting factor, factor 3, which interacts with calcium, and factor 7 here to form a tissue factor complex, which then activates factor 10. And we saw in the video that factor 10 there's a 1 to 10 ratio here, right? So for every factor 10 that is activated, that affects 10 times the thrombin. So thrombin is in an inactive form called prothrombin, and then when it interacts with the factor 10, it forms thrombin. Thrombin is what catalyzes the fibrinogen molecules in the plasma to convert to fibrin, and fibrin is what forms the mesh, right? That helps attract more and stabilize more platelets here to stop the bleeding. Now, also, when we have a tissue break, we have intrinsic chemicals that are released. So, in other words, these platelets themselves, which are part of our blood, are going to release things like platelet factor 3 and some other clotting factors, which also interact with calcium, which interact with some other factors. Factor 8 and factor 9 here, forming that factor 10 activator complex, which then feeds into the common pathway and continues to generate the fibrin, which forms the mesh. And then the video got in a little bit into the breakdown of this, right? The reverse process that basically breaks all this back down once this tissue has been healed over and reverses this process. So the blood clotting cascade is pretty complex. There are many factors involved, and just to give you an idea, here are all the different factors involved with clotting a protein and their name. So for example, we looked at factor 1, which is the fibrinogen. We also looked at factor 2, which is the prothrombin, right, the inactive version of thrombin, which then activates the fibrinogen to become fibrin. So all of these tissue or all of these clotting factors work together in order to form blood clots. If we have one of them that isn't working properly, then that's going to impact the whole, what we call the clotting cascade. So there are different disorders of blood clotting that involve some of these different clotting factors. So, for example, one clotting factor disease that you may be familiar with is hemophilia. And so and there are even different types of hemophilia depending on what clotting factor is impacted. But no matter the type of hemophilia, basically what happens is, so here's our normal coagulation, right? We have a wound, we have coagulation factors in the blood coming together to clot the blood, right? Normal blood coagulation. In hemophilia, certain coagulation factors are missing from the blood. And so if we're missing just one of those, that whole cascade of events can't happen, and we don't get the clotting that we normally would. We don't get stoppage of bleeding in as quick of a time. And so people with hemophilia are at risk for increased bleeding because their blood doesn't clot properly. There are also some disorders with having blood clots form when they shouldn't form. So hemophilia is clots not forming when we need them, but we also sometimes get blood clots forming when we don't need them to form. So, for example, when we have a buildup in blood vessels, that can trigger that intrinsic pathway to form blood clots when there is actually no bleeding. So a thrombus is basically a blood clot that forms in an unbroken vessel. So this is a blood clot that's going to form and it can block blood flow. So you can see up here in this first picture, we have a patient that has definitely swelling edema happening in one of his limbs and not the other. And this is due to a deep vein blood clot that's blocking venous return. So blood's coming down to his feet, and then when it's trying to circulate back up to the heart, it's getting blocked by a clot in a blood vessel, causing the swelling that we see here. Now, an embolus is more dangerous. This is when that thrombus starts to move. So when that thrombus breaks free from the vein and begins to travel through the system, it can cause problems, right? So if that blood clot then works its way up to the heart and gets stuck there, it can cause a heart attack. It could cause a burst blood vessel, an aneurysm. If it gets stuck in a blood vessel in the vein and blocks blood flow, it can cause a stroke. And so an embolus, moving a blood clot, is much more dangerous. Blood clots can be broken up different ways: medication, blood thinners, sometimes surgery. Depends on where the clot is located, and if it's moving or not, and the history of the patient, but they can be deadly. Okay, our next and last topic for blood is going to be blood typing. So that's what we'll take a look at during our last mini lecture on blood.